Welcome back to part 5. In this section, we're going to go over the tSNE and UMAP plotting of the data. So taking off from the FlowSOM clustering, we are going to start by doing the heat maps of the data, and this is just a rather crude analysis of the data based on the median fluorescence intensity in each marker by cluster. So that's this section here, starting with plot heat maps and the analysis script. I'm going to select all of this, run it, and then look at our outputs. And we should have here in heat map 3 different plots, one based on cluster, the other by condition, and the third one by individual sample. So if we look first at cluster, we see something sort of similar to what we saw before with the histograms, where we can now have our cluster names showing up here. So our naive CD8s have red, so high levels of CD8, and high levels of CD62L here. And then, for instance, our CD8 Chi67s have high CD8. And we see now there's a problem here in the naming, in that Chi67 isn't coming up on this side. And what you can see by looking closely is that there's more bars here than there are names, which is because the names in the text is too big to fit with the number of markers that we actually have in the data set. So we need to change the plots in a second, which we will do. If we look at the next one, we can see this is by condition. So we have our 4 conditions, our 4 groups, and we can see that our two, we have a very crude dendrogram here across the top, which is saying that our 2 blood samples are closer together than the spleen, and similarly our 2 spleen samples are grouping together. And it's showing this, the heat mapping across our groups. And so this would basically tell you that in theory, say CTLA4 is highest in the wild type blood compared to the spleen, which, given that we're working with the entire T cell population rather than, say, Tregs, isn't particularly meaningful. But this might be helpful in some cases. One that can be quite useful is the heat map by sample. And this will tell you how similar your samples are. So by looking here, you can see that, again, all of the blood samples are on one side and all of the spleens on the other side. So clearly the blood and spleen are distinct. What's also interesting is that most of the amphiregulin knockout samples have clustered together. So perhaps there is actually a biological difference in the absence of amphiregulin. And you would say, all right, let's zoom in here and see what it is. And it's this line here is driving some of the difference. And CD4 is also driving some of the difference. Over here in the spleen, it's not so clear cut. We have the samples sort of spread out a bit. It's not quite so clear if there's actually a difference between these two. What can be quite nice is if you get clear grouping of samples in one condition versus the other, you feel relatively confident that there is a difference that is consistent across all samples in your condition. Right, so let's fix this issue that I mentioned earlier with the labeling being off compared to the scaling of the plot. To do that, let's close this. We go back to our parameter file. And we go down to heat map parameters. So here we have several parameters. You can change the color or the palette that it's using, the scaling of the colors. Probably what you're going to need to change is either the font size, which you can make smaller, or more commonly, the width and height of the figure. So if you have a bigger figure like we do with many parameters in your data set, you need to make the bars here bigger rather than trying to make the text smaller. Otherwise, you won't be able to see the text very well. So let's change this from 800 to 1200. 1200 is generally a good size. So having 1200 for both will obviously make it a square heat map. So because we've changed those values, we're going to enter them by doing Control Enter, and also save this. And then we can rerun the make histogram command. and we should get new histogram heat maps, sorry, with, you can see this one's just been remade. And now we have space for each marker listed along the edge. So we can now see that in addition to CD4, there's a change here potentially in CD103, possibly in Chi67, and ICOS. So now we can really see what each of these markers is contributing to the data set. Again, this is relatively crude because it's based on median fluorescence intensity. So it's not the greatest, and it's not going to be that helpful for a clustering analysis. Right, so let's proceed from onward and look at the histograms. The histograms are going to plot the clustering that we did earlier. So here we get 3 histograms. Histogram by assay. This is not the most useful graph. This just plots the total number of cells in each group, in each cluster. So you don't have any error bars because there's only the total group, but it shows you that most of the cells are naive CD4s and CD8s. The next plot is histogram by cluster with the change in frequency between the different groups. So we can see that there's relatively little change in log-fold change in the larger groups because they're pretty numerous and therefore the numbers are more robust. And the smallest groups have much greater variability. And what I tend to use most is the average frequency, which this shows us that in the blood samples, we have a difference in the frequency of CD8s and CD4s, depending on the presence of amphiregulin or not. So in both cases, the spleen and the blood, we have a shift in the balance of CD4s and CD8s, for instance. And you can see that down here in these other charts, you also have changes. And again, this part over here on the right-hand side, you can change the colors palette in your parameter file. Here in your histogram parameters, you can change the size and the text of the histogram. And if you want to have the groups show up in a different order, for instance, if you want the spleen first rather than the blood, you change that in your parameter file up at the top here where you define the order of the conditions. Right, the next step is to calculate the t-SNE representation. We're just going to collect, do all of this and hit enter. And that may take a while depending on your computer and how many events you have. All right, so that has completed. As an aside, if you are plotting large data sets of t-SNE, you will probably be better off running this in R rather than RStudio. It just tends to fail less. Sometimes it gets caught up in loops. All right, so the next step is to plot the t-SNE convergence. And we can have a look at that quickly. So you should see this nice curve where the first couple steps are rather crude in the t-SNE algorithm, and then you get this nice smoothing off of the data. And if this looks like it's still dropping off really rapidly, then you want to run more iterations. So for instance, if we had run only 1,000, which is sort of the default in most flow programs, you'd see it will look like this, and it only recently started to level off. And really by increasing the number of iterations, we get usually better t-SNE representations. Although obviously the longer you run it, the more it's going to take and the less you're going to gain. Right. So we're going to now plot the rest of the t-SNE representations. And this will again take a minute while it generates all the graphs. and you can see them coming here. And what it's going to do is plot a t-SNE diagram for each marker that we used to generate the t-SNE, and also one with the FlowSOM clusters in overlay. These all event plots are useful for understanding where your markers are on the t-SNE. So here's our t-SNE plot. You can see we have actually pretty nicely defined clusters. And that's because we're using markers that are really well distinguishing, like CD4 and CD8 in our plot, which is driving clean separation. So you have CD8 clusters down here, whereas these other ones up here are probably CD4s or gamma deltas. If we look at, say, another marker, CD25, you see that up here, that's probably a regulatory T cell population. Whereas, say, CD44, which is an activation marker expressed in all the subsets, is sort of split between all of the different major populations. And you can go through this for each marker and understand how it's contributing to the t-SNE or whether it is. So for instance, CD95, which is in most cases relatively low expression and only really highly expressed on some activated Tregs or thymocytes, isn't really contributing much to the t-SNE. And you can see that here, it's just sort of all over the place and there's no highs and lows. So you could, for instance, run the t-SNE again without that marker if you wanted. It doesn't really matter much, it's just not contributing either way. Here, this one, we have our FlowSOM clusters, and you can see, again, that our naive CD4 and CD8s are taking up more space in the diagram. And because on t-SNE, the events can't occupy the same space, that also shows us how much of each cell type there is visually. Right, so it's done plotting all of those. So you also get these side-by-side plots where you can see, first off, that the spleen and blood are occupying completely different halves of the plot. So they are really different samples. And you can see whether there is actually a difference between the different samples. So for instance, the wild type and epiregular knockout spleen plots actually look very similar. The cells are in all of these different places. And we'll get to this a bit more with the t-SNE diff function. But really you can see already that these two plots for the blood are quite similar shapes and the spleen are quite similar, whereas there's a big difference between the spleen and the blood in general. So that's your t-SNE plots. If you find that this is just sort of a mess and there's not really any clear groups anywhere, you may need to redo the compression of the negatives, or think about how to better set up your experiment in terms of what cell populations you work with, or how many iterations you need to run, or what markers you're using for the analysis. Some cell types like Tregs, you will basically just get one cluster that sort of roams off here and there because they're all so related. All right, we're going to proceed with the UMAP, which is basically the same. So I'm just going to select a couple sections all the time. So we'll run the UMAP and then plot all of the representations at once. And again, this will take a couple minutes, so we're going to take a break here. All right, so our UMAP plotting has finished. So we're going to have a look at that. So similar to our figure t-SNE, we have a figure UMAP. And here we have, for instance, let's look at our CD4. And you can see that the UMAP plots really look rather different. They tend to compress the data a lot more in the space, and you get this bigger separation between the clusters. And so we can go through and have a look at a couple of these. And it's a different representation of the data. In some cases, you may prefer one or the other. It's more of an aesthetics thing most of the time. Sometimes one of them performs better at the other in putting similar cells together in difficult data sets. This one's pretty simple, so clearly the clusters are quite well separated. Right, we're going to proceed with the tSNE diff. And here, it's pretty important to set some of the parameters in your parameter file because this is really a heavy calculation for your computer. It will probably crash on most computers if you try to run more than about 100,000 events. So you need to go down to the cross-entropy test parameters. So this part here, instead of putting this as null, I would recommend in general that you put 100,000. That way, if you are analyzing more than 100,000 cells in the tSNE and UMAP and FlowSOM, it will only take the first 100,000 for the analysis here. That does limit your power somewhat to differentiate the samples, but that's okay in most cases. It's going to speed up the calculation quite a bit. In my case, I should only have about 32,000 events, I think. It depends on how much RAM you have on your computer. The other part that you may want to change would be the type of test that it's doing. So KS, Kolmogorov-Smirnov test is the better one to use. The other option is to put rank and median here. This is not as powerful, but if you're having difficulty in some cases trying to figure out if it really is a statistical difference, you can run it both ways and see which works better. There are certain instances where rank and median is more appropriate than KS statistics, but there are not very many of those. All right, so let's save that. Again, we need to enter this. For now, in order to speed this up, I'm just going to limit it to 10,000. All right, I'm going to calculate the cross-entropy probability for the tSNE. And because I've set such a low number, it's pretty fast. Let's have a look at that. So by restricting it to a lower number of events, we restrict the power of the test as well. So you get three outputs here. One, you get these CDF probability curves, and you can see when the curves are jumpy like this, it means that there's not really enough data to make them smooth. So especially for our less frequent clusters, you have jumpy data when I've restricted the data such. And this is looking at the clusters. So we're seeing the difference between each cluster. So the median is this black line, all of the events, and you can see how much each one diverges from that. And then you have the difference when there's that each cluster is statistically different from the other clusters. And so that's what we've run so far, is just looking at the clusters. And we'll run again in a minute for the samples. And so this is a dendrogram based on the cross-entropy. For the clusters, you can see that naive CD4 and CD8 are together, whereas we have a grouping of our other cell types as such here. You can rotate this if it's easier to see it that way. I don't use the cluster ones very much, but it might be relevant for specific projects. And then you can hear you have your actual p-values, and this tells you the p-value and your adjusted p-value. So an adjusted p-value of 0 is really, really low. So obviously, naive CD4s and CD8s are very, very different because they have one has CD4 and one has CD8, and it's a very yes-no difference. You're not going to confuse the two. So you have here a comparison for each cluster versus all the other clusters, and whether the p-values are telling you whether there's a statistically significant difference, bearing in mind that I have restricted the analysis to only 10,000 events. And if we up the analysis to the entire population, you would see much lower p-values in most cases, because we would have a larger sample size to work with. Right, we can do the same thing for the UMAP. And as I skipped over it earlier, we also have the cross-entropy t-SNE per sample. So we'll run that. All right, so now we have the sample results. So here's our dendrogram per sample. So this is based again on the cross-entropy. And we can see that our blood samples are mostly grouping up here, with the exception of one spleen. And our spleen samples are mostly grouping down here. And if you compare this to how the cells were distributed on the dendrograms that we got by the heat map, you can see there's some differences. So we can rotate this one the same way and look at these side by side. And then with the heat maps, you can see that we got very nice separation of the spleen and blood being the primary branching in the tree. And that sort of makes sense if you look at the actual t-SNEs. So blood is clearly all on one side, spleen is clearly on the other. So you would expect that to be the primary division. But the dendrogram on the cross-entropy test sort of is different in a way that I don't fully understand, but it looks at the local structure of the data rather than the broad structure of the data. So you don't always get the same results. Right. So let's run the last bit of the UMAP cross-entropy. And then have a look at that and see if that looks any different. So here we have our cluster plots. And here we have one for condition. Here are the individual samples, the CDF curves. So you can see that the wild type spleens are all coming up on this side. So they're actually differing a bit from the other samples. And here we have our dendrogram of the clusters. Looks quite similar to the t-SNE dendrogram of the clusters. Here we have a dendrogram of our conditions. Which I didn't run. So we can compare this side by side, and you can see that the UMAP one on the left has same structure as the t-SNE one. And again, sort of a similar setup here, some set differences in the structure of the trees. Again, neither of them looks exactly like the dendrogram that we got with the heat maps. And then finally, probably what we're more interested in is looking at the p-values to compare the plots. So we can do that for the t-SNE or the UMAP. So let's have a look at the UMAP. And we probably care most about the conditions, which is the overall groups. So is wild type blood statistically different from average regular knockout blood? And the adjusted p-value is 0.01. So it is, that's not a big difference though, particularly when you look at, say, the comparison between wild type blood versus wild type spleen. We can see the p-value is 10 to the negative 9th power. So each of these p-values You can see the wild type spleen and F regular knockout spleen here are coming up as really hugely different by p-value. Again, this is something you'd want to run with as many cells as you're capable of running on the machine, and have a look at the data that you're getting with both conditions and see if it makes sense. So here you can see we're getting something similar for the wild type spleen versus amphiregular knockout spleen, 10 to the minus 12, 10 to the minus 14. So they probably are different in some way. Then you need to go back and look at your t-SNE plots and your bar charts and your histograms to try to figure out what that difference is. If you feel like there's something a little bit odd about your results with the t-SNE diff, what you can do is go back and rerun it. You can set a different seed and run it again. Or you can run the whole script again with by setting the seed here to a different number. You can run the whole thing again, get a different set of t-SNEs, a different set of UMAPs, and then you will run the t-SNE diff on those new distributions of the data. And if you get similar p-values, you'll know that really it's probably a real effect, that there is a difference in the phenotypic distribution of the cells. All right, and that's basically it. Let's touch on a couple things with quickly about how to change the data output in terms of the graphics. So we looked earlier at changing the heat map to get the scale on screen. You can do the same thing with your dimensionality reduction figures. So your t-SNE and your UMAP plots. So you have all of these different parameters. Each one changes something. So the legend titles, the legend label size, these fonts. Probably the more important one in most cases is going to be doing something like changing this line here. So for instance, right now, if I look at my t-SNE plots, I have all of them in one row, which is being specified here, n row number of rows equals 1. If I want to have 2 rows, so I want to have the wild type spleen down here and the F-regular knockout spleen down here, so a square, I would set this to be 2. If I want the plots to be larger or smaller, I change these here. So right now they're set to be 3, which I think is in inches, and I can change this to be 2. If I then go to my script and say plot t-SNE representations, it should replot all of these plots with those new parameters. And so let's keep an eye on, say, this one where we have, see this plot right now with this text size has been chosen to be sort of appropriate for this. And because we've changed the size of the plot, we'll now see how that, without changing the text, we'll see how that works out. So now you can see my new plots here, which are now designed the way I wanted with 2 rows instead of 1 row. All right, so that's done. So now the text is larger with respect to the plot. That may be useful to be able to change that. The other factors you can play around with if you want, but those are the main ones that I use. So I think that's enough for now. And probably if there are a bunch of questions, I'll do a follow-up. So thanks.